Okay, so agarose gel electrophoresis, DNA digestion with restriction enzymes, and its application to DNA separation.
So gel electrophoresis, agarose gel electrophoresis is a process in which the electricity is used to separate DNA fragments based on size, and we can see this commonly found in investigative crime scenes and other sorts of applications dealing with comparisons of DNA or genes. So whenever you're preparing your gel electrophoresis, you first need to prepare your gel, and in order to prepare your gel, you need agarose and you need a buffer. And depending on the concentration of your agarose, you may want to vary the percentage. So usually around 1 to 5% agarose. Once out of the refrigerator, the gel will harden, and it will actually turn into a gel, and the tray will then be set into a salt solution. And before messing with anything else, you will need to implement blue tracking gel to the DNA sample so that you can see it later on in the gel electrophoresis procedure. And then you need to insert the DNA samples into the slots that you made in your gel from whenever you set it up in the first place. So there needs to be a chamber in which the electrophoresis takes place. So then you will place the tray that you have prepared into that chamber, and you'll add the buffer solution on both sides of the gel box to where it's completely covered. And then you will then place the slots at the negative end of the chamber and then turn on the power supply for the electrophoresis process to start. So a part of the DNA backbone is phosphorus, and phosphorus has negatively charged oxygen molecules. Therefore, whenever the electrophoresis chamber is turned on, the negatively charged DNA will move across the gel that we created to the opposite side of the gel towards the positive end. And then smaller strands, since they weigh less, will move farther than the longer strands that weigh more.
So just to start off, like how the DNA becomes into those fragments, DNA is processed before placed in the gel by restriction enzymes that are also called endonucleases. These are often called molecular scissors that cut at a specific sequence. These sequences are often the same way they are forward and backwards, because DNA is double-stranded and it's like a complementary base pair. So they actually will be the same, as you can see in the second picture, where they, even though they are kind of flipped, they're actually the exact same. Yeah, so each enzyme is actually specific to one code, one sequence. So it only cuts at that one location and won't cut anywhere else. Yeah, could we get the next slide? Here's another picture of just like how in like this big sequence, even though there are other bases, they only cut at the specific place. So here we have like a chromosome, but also like the DNA being like unwound across it. We see that it's very, very, very long. Human DNA has over 3 billion base pairs. That's actually like I think it's like a little bit over 5 feet in every single cell. But the enzyme will go across each of these bases and then recognizing where the sequence is to cut. So that's how we get these different segments of different sizes. So each individually in a different DNA and a different DNA sequence will have varying amounts of the pieces.
Cool. Once the gel has finished running and gel electrophoresis, it's time to analyze the data, and we need to be able to visualize the DNA fragments. So this picture on the right shows what the data would look like once the process is completed. The lines that are visible are called bands. These bands show how many base pairs there are in the DNA analyzed. As you can see in the photo, there is a DNA ladder, which is that first column right there. It is compared to the other ones, and those can be labeled whatever, depending on what you're analyzing. The number on the side represents the amount of base pairs in the DNA samples. Now that you know about the gel electrophoresis, here are some real-world applications. Some are DNA profiling, paternity tests, analysis of antibiotic resistance, analyzing genes associated with illnesses, fingerprint tests, and be able to separate DNA fragments to clone specific bands of DNA. One problem with using DNA separation in criminal investigations is that human error is a big aspect, and this is a fragile process, so you can mess anything up, which could be dismissed in a trial. Okay, so